what if the wrinkle cream is fighting the wrong battle? Discover 13 collagen plants that instantly reverse aging effects. It sounds like a headline designed to make you click. The title promises instant reversal. Here's the reality. Some plants deliver immediate protective or signaling effects, but structural repair and visible rejuvenation build over weeks to months when you use them consistently. That caveat matters because it separates hype from helpful action. There are real, measurable things plants do the moment they meet your cells, and there are slower processes, building new, well-formed collagen fibers that require time, substrates, and repeated signals. Let's start with the biology briefly but clearly. Collagen is not a surface thing. It's a structural protein assembled deep in the connective tissue by fibroblasts, the little cellular craftsmen responsible for laying down the scaffold of skin, tendons, and much of what keeps tissue resilient. From our mid-20s onward, fibroblast output wanes, and enzymes called matrix metalloproteinases, MMPs, think of them as a demolition crew that goes into overdrive when inflammation or UV exposure spikes, begin cleaving existing collagen more than it's replaced. For collagen to be synthesized correctly, fibroblasts need amino acids, yes, but also key cofactors. Vitamin C is essential for prolyl and lysyl hydroxylases to stabilize the collagen triple helix. Certain polyphenols and proanthocyanidins help cross-link and protect fibers. Fatty acids like palmitoleic acid can integrate into membranes and influence skin barrier health. And antioxidant systems such as NRF2 reduce the oxidative signals that trigger MMP activity. Creams and serums can hydrate and temporarily plump the surface, but they seldom get deep enough to change fibroblast behavior or meaningfully suppress the internal demolition crew. That's where plants come in. Many botanicals provide raw materials, cofactors, and signaling molecules that tell fibroblasts to produce, protect, and stabilize collagen, sometimes within hours at the molecular level, sometimes over repeated exposures across weeks and months. If you want a one-sentence map before we go plant by plant, some of these foods give the essential vitamin C your cells need to build stable collagen. Others calm inflammation and blunt MMPs. A few supply specific lipids or proanthocyanidins that help collagen fibers stay intact. If this kind of evidence-based nutrition interests you, hit like and subscribe. We'll break down the research every week. Now, we'll count down from 13 to 1. Each plant gets the same short treatment, what it chiefly contributes, the mechanism in plain language, how to use it practically, and a short qualifier so expectations stay realistic. 13. Guava, the vitamin C powerhouse that feeds fibroblasts. Guava is one of those tropical fruits that reads like a supplement label, extraordinarily high in vitamin C per serving, along with a mix of flavonoids and carotenoids. Its chief contribution to collagen is straightforward. It supplies the ascorbic acid fibroblasts need as a cofactor for prolyl hydroxylase and lysyl hydroxylase, the enzymes that chemically modify collagen strands so they fold into a stable triple helix. Without adequate vitamin C, the helix remains underhydroxylated and less mechanically resilient. At the molecular scale, that supply can have near immediate benefit. Once vitamin C is available in the local tissue environment, the enzymatic machinery for collagen maturation can function more efficiently. In practical terms, this means the raw materials you already have are more likely to be laid down correctly. For people low in dietary vitamin C or those whose intake is inconsistent, adding guava, fresh or as a high-quality frozen puree, can shift the balance quickly toward better collagen synthesis. Practical tip: slice guava into smoothies, salads, or eat fresh. If fresh guava isn't accessible, look for minimally processed frozen puree or whole fruit packs so you get the vitamin C matrix along with fiber and polyphenols. Because vitamin C is water-soluble, Pairing high vitamin C foods with meals is simple and safe for most adults. Quick qualifier. While guava supplies an essential cofactor and may prompt more efficient collagen processing, it is not a magic bullet on its own. Structural rebuilding still depends on repeat, balanced intake and overall protein and micronutrient sufficiency. 12. Acerola, concentrated vitamin C in a handy form. Acerola cherries pack vitamin C in even higher concentrations than many common fruits and are often used in freeze-dried powders and whole fruit supplements. Like guava, acerola's key role is supplying ascorbic acid to support the hydroxylation reactions that stabilize newly synthesized collagen and prevent misfolding. Acerola also carries a matrix of polyphenols that may protect vitamin C from rapid oxidation and provide complementary antioxidant effects. In research contexts and in traditional use, acerola shows up as a rapid way to boost systemic vitamin C levels. Practically, that means a serving of freeze-dried acerola powder stirred into a smoothie or water will increase the availability of the cofactor fibroblasts need within hours. The immediate effect is biochemical, better activity of key enzymes. The visible effects, however, require consistent intake and the body's own tissue-building timeline. Practical tip. Consider freeze-dried acerola powder as a convenient way to add vitamin C without excess sugar. It keeps the natural matrix of the fruit. Start by adding a small spoonful to morning smoothies or yogurts. Quick qualifier. If you're using an acerola supplement, choose whole fruit or minimally processed formats rather than high-dose isolated ascorbic acid unless advised otherwise. And keep in mind that increasing vitamin C can only help if other building blocks and lifestyle factors support tissue synthesis. 11. Red bell pepper, vitamin C plus carotenoid armor. Red bell peppers are probably already in your shopping cart, but they deserve a starring role here because they combine high vitamin C with carotenoids such as capsanthin and beta-carotene. That pairing supplies both a cofactor for collagen assembly and lipid-soluble antioxidants that attend the collagen matrix, particularly at cell membranes and within lipid-rich regions of skin. Mechanistically, the vitamin C in red pepper supports enzyme-driven stabilization of collagen helices, while carotenoids help quench lipid peroxidation and reduce oxidative signals that would otherwise activate MMPs. In short, they both help make collagen and help preserve it once made. You'll also get fiber, folate, and a suite of minor nutrients that support general tissue health. Practical tip. Eat red peppers raw or lightly roasted to preserve vitamin C and carotenoids. Add them to salads, stir-fries finished with a drizzle of healthy oil, or blend into sauces. The fat helps carotenoids absorb. Quick qualifier. Vitamin C in peppers is abundant but not limitless. A varied diet with multiple vitamin C sources is still advisable for consistent fibroblast support. 10. Korean ginseng, a stimulant for fibroblasts and hormonal balance. Korean ginseng, Panax ginseng, contributes a different kind of support. Ginsenosides, the active saponin compounds, which research suggests can modulate fibroblast activity and blunt stress-related pathways that lead to tissue breakdown. Chronic elevated cortisol and stress signaling can indirectly promote MMP activity and impair repair. Ginsenosides appear to buffer some of that signal, and in cell and animal studies, enhance markers of fibroblast function. That doesn't mean ginseng replaces stress management, but it can be a useful dietary signal. If fibroblasts are being repeatedly told to slow down by inflammatory or hormonal cues, a compound that modulates those signals can free them to do their job more effectively. In practice, people report both subjective and biochemical improvements in skin resilience with regular use of standardized ginseng extracts, though individual responses vary. Practical tip: Korean ginseng can be consumed as a tea, tincture, or standardized extract. If you opt for an extract, look for standardized preparations and start conservatively, especially if you take medications or have blood pressure sensitivities. Ginseng can interact with certain drugs and may not suit everyone. Quick qualifier. The evidence supports ginseng's role in modulating fibroblast signaling and stress pathways, but it's not a universal solution and should be used thoughtfully. 9. Rosemary, antioxidant protection and anti-inflammatory signaling. Rosemary contains carnosic acid and rosmarinic acid, two compounds with potent antioxidant and anti-inflammatory properties. These molecules help reduce the oxidative triggers that activate MMPs and other catabolic pathways. In other words, rosemary acts like a protective shield that lowers the red alert signals in tissue, limiting unnecessary demolition of collagen. There's an additional benefit. Carnosic acid has been shown in lab studies to support cellular stress response pathways that favor repair and resilience. When combined with other antioxidants, rosemary's phytochemicals contribute to a tissue environment where fibroblasts can rebuild rather than merely patch. Practical tip. Use fresh or dried rosemary in cooking, infuse in oils, or choose a culinary rosemary extract for concentrated applications. As with other plant antioxidants, pairing with dietary fats can improve absorption of certain lipophilic constituents. Quick qualifier. Rosemary supports antioxidant defenses and inflammation modulation, but should be seen as one node in an overall anti-catabolic strategy rather than a lone protector. 8. Turmeric, a targeted inhibitor of enzyme-driven breakdown. Turmeric's star compound, curcumin, is well known for its ability to reduce inflammation and, importantly for collagen, to inhibit UV-triggered MMP activation in laboratory studies. When skin or connective tissue is exposed to pro-inflammatory triggers like UV light, a cascade can lead to increased MMP expression. Curcumin acts at several points along that cascade, reducing signals that tell the demolition crew to swing its hammers. There's a practical catch with turmeric. Curcumin is poorly absorbed on its own. Fortunately, traditional culinary wisdom and modern research converge, pairing curcumin with piperine, a compound in black pepper, and dietary fat markedly improves its bioavailability. Some clinical studies referenced in the literature use standardized curcumin extracts, 95% curcuminoids, to ensure consistent active content, which is useful when aiming for research-like effects. Practical tip. Include turmeric in meals cooked with a source of healthy fat, olive oil, coconut milk, and a pinch of black pepper. Or consider a quality standardized curcumin supplement if you seek concentrated intake. Quick qualifier. While curcumin can blunt MMP activation and inflammatory signaling, it does not instantaneously rebuild lost collagen. Its strength is in protection and signaling moderation that supports longer-term repair. 7. Green tea, EGCG, the enzyme whisperer. Green tea's most active polyphenol, EGCG, suppresses MMPs and neutralizes oxidative triggers that lead to collagen breakdown. It's a two-pronged approach. EGCG reduces the signals that call the demolition crew, and it also promotes an antioxidant environment that favors collagen integrity. In various experimental systems, EGCG has been shown to modulate gene expression related to extracellular matrix homeostasis and to protect cells from UV-induced damage. Drinking green tea or using matcha delivers a steady stream of catechins that work systemically and locally. Because green tea is widely consumed and generally well tolerated, it's an easy way to add MMP-inhibiting phytochemistry into a daily routine. For those who prefer concentrated forms, standardized extracts provide a higher, more consistent dose of EGCG, but even routine brewed tea has meaningful activity. Practical tip: enjoy green tea daily, brewed rather than boiled aggressively. or include matcha in smoothies for a concentrated catechin hit. If you choose an extract, opt for products standardized for EGCG content. Quick qualifier. Green tea supports MMP suppression, but its effects are part of a mosaic. Drinking tea helps, but it's most effective combined with a variety of other plants that supply cofactors and stabilize collagen fibers. At this midpoint in our countdown, a pattern emerges. Some plants are raw material donors, guava, acerola, red pepper supplying vitamin C. Others are protective agents that blunt destructive signaling, rosemary, green tea, turmeric. And some modulate the cellular climate to favor repair, ginseng. The real advantage comes when you combine these functions, when you feed fibroblasts the cofactors they need, while simultaneously pacifying the biochemical alarms that would otherwise accelerate breakdown. Before we move from these initial 7, consider a practical short-week plan to fold them in. One day could center on vitamin C-dense fruits, guava in the morning smoothie, red pepper snacks midday. Another day could emphasize anti-inflammatory herbs and spices, rosemary and turmeric in soups and dressings, with green tea in the afternoon. A third day might include a cup of Korean ginseng tea or a modest extract, serving to support hormonal balance. Rotating these plants through the week ensures your fibroblasts repeatedly receive both the building blocks and the signals to stay active. A quick micro hook. Next we move from vitamin C, heavy hitters and enzyme blockers into traditional powerhouses and targeted stabilizers. Plants that bring unique molecules like proanthocyanidins, palmitoleic acid, acemanin, and the unusual polyphenol architectures found in amla and rose hip. These are the botanicals that help collagen hold together once it's made, and they come with cultural histories that modern research is beginning to validate. 6. Amla, whole fruit vitamin C with a stabilizing matrix. Amla, or Indian gooseberry, has been used in Ayurveda for centuries as a rejuvenative. Modern analysis shows it's exceptionally rich in vitamin C, but importantly, the vitamin appears bound within a dense polyphenol matrix. This matrix may slow oxidation of vitamin C, offering a more durable supply of ascorbate in the tissue environment than isolated vitamin C alone. Practically, amla's combination of vitamin C plus tannins and other polyphenols supplies both the cofactor fibroblasts need and the protective entourage that limits its breakdown. Mechanistically, that means amla can contribute to both increased enzymatic hydroxylation of collagen and ongoing antioxidant protection in the extracellular space. In some cell studies and traditional use reports, amla preparations are associated with improved markers of skin resilience and reduced oxidative stress. Practical tip: use amla powder in smoothies, chutneys, or as a tincture. Fresh amla is ideal when available. The whole fruit approach preserves the polyphenolic scaffold that appears to stabilize vitamin C. Quick qualifier: effects from amla are supportive and cumulative. It's a potent addition to a collagen-supportive diet, but not a standalone cure. 5. Rose hip, vitamin C plus gopo for joints and skin. Rose hip brings vitamin C together with a specific set of oligosaccharides and polyphenols, sometimes referred to under trade names like gopo. These compounds have been studied for joint comfort and for supporting connective tissue health. The vitamin C in rose hip supports collagen synthesis, while gopo-like molecules appear to support joint-specific matrix stability. For skin, that means rose hip provides both building blocks and extracellular protection that can help tissue retain elasticity and resilience. In some clinical contexts, rose hip preparations have been associated with improvements in joint-related outcomes and skin elasticity markers. Mechanistically, the combination of vitamin C and unique soluble polyphenols helps both production and maintenance of collagen fibers, especially in tissues under mechanical stress. Practical tip: Consume rose hip as a tea, whole fruit jam, or look for standardized rose hip extracts that emphasize the whole fruit matrix rather than isolated components. Quick qualifier. Rose hip is particularly useful for people concerned with connective tissue load and joint-skin interactions, but individual responses vary and more research continues to refine optimal forms. 4. Grapes. Proanthocyanidins and resveratrol for fiber stability. Grapes, and especially grape seeds and skins, are rich sources of proanthocyanidins, polymeric flavonoids that can stabilize collagen fibers and inhibit enzymes that degrade extracellular matrix. These compounds bind to collagen and can increase its resistance to enzymatic breakdown in laboratory models. Resveratrol, present in grape skins, also activates pathways associated with cellular resilience and longevity, though its systemic bioavailability is complex. The net effect is twofold. Proanthocyanidins act as molecular cross-linkers that make collagen less susceptible to breakdown, and resveratrol-like molecules support cellular stress management. In practice, grapeseed extract standardized for proanthocyanidin content is often used in studies aiming to protect matrix integrity. Practical tip: eat whole grapes for skins and polyphenols. Include red or purple varieties regularly, and consider grapeseed extract standardized for proanthocyanidins if you want more concentrated support. Quick qualifier: while proanthocyanidins help fiber stability in models, clinical outcomes depend on dose, form, and individual metabolism. Whole food intake plus varied plant polyphenols is a balanced approach. 3. Aloe vera, oral and topical hydration with asemanin support. Aloe vera is familiar as a topical soothing agent, but oral preparations of inner fillet gel contain asemanin, a mucopolysaccharide that may support hydration and matrix maintenance internally. Human trials have reported improvements in skin elasticity and moisture with oral aloe products, suggesting that aloe's effects are not purely surface level. For collagen support, aloe contributes to a hydrated extracellular environment and provides molecules that may support fibroblast health and mucopolysaccharide balance. Mechanistically, by improving hydration and providing specific polysaccharides, aloe can create tissue conditions favorable to collagen assembly and functional elasticity. Topically, aloe hydrates skin and soothes inflammation, both useful for reducing triggers of MMP activity. Practical tip: choose inner fillet oral aloe gel products or, when using fresh leaf, carefully prepare to separate the inner gel from latex-containing layers. For topical use, quality aloe gels can be part of a routine to maintain hydration. Quick qualifier: not all aloe products are equal. Standardized inner fillet preparations better match the formats used in research, and those with added sugars or contaminants should be avoided. At this point, we've covered vitamin C heavy hitters, enzyme modulators, stabilizers, and both oral and topical hydrators. The remaining plants in our countdown, broccoli, sea buckthorn, and the others we haven't yet reached, bring mechanisms like NRF2 activation, unique fatty acids, and concentrated polyphenols that further round out the strategy. These final plants are the ones that link metabolic signaling with structural integration. They're where tradition and modern studies most directly intersect to give a practical, actionable path forward. We'll pause here on the narrative arc because integrating these foods into real life raises a set of practical questions that people frequently ask. How to combine these plants across a week, what forms are most bioavailable, how to handle interactions or contraindications when you're on medication or pregnant, and how to prioritize if your budget or taste limits you to a few choices. Those decisions shape outcomes as much as the individual plants themselves, and the next section will translate mechanisms into a simple, repeatable plan you can start today. choosing 4 to 6 staples, pairing for absorption, and rotating seasonally, while also addressing common safety considerations and how to spot quality in extracts versus whole foods. But before we lay out that actionable roadmap, there's one more scientific hinge to understand: how the cellular signaling changes immediately when these compounds arrive. In many cases, molecular events begin within hours. Antioxidant defenses upshift, MMP expression drops, and cofactors for hydroxylation become more available, creating a biochemical environment primed for repair. Those immediate shifts are real and measurable, and they're what lets us say the plants can instantly influence the processes that lead to aging. The structural consequences, firmer, visibly rejuvenated skin, require the repeated, day-to-day presence of those signals and raw materials. Next, we'll finish the countdown with the last 3 plants, and then move into a clear, practical rotation you can implement without overcomplicating your life. We'll also unpack bioavailability tricks, like turmeric with black pepper and fat, broccoli preparation methods to preserve sulforaphane activity, and how to evaluate product standardization when a concentrated extract makes sense. 2. Broccoli, the NRF2 switch that rallies cellular defenses. Broccoli isn't just a side dish, it's a biochemical signal. The key player here is sulforaphane, a compound formed when broccoli's enzyme myrosinase encounters glucoraphanin after chopping or light chewing. Sulforaphane activates NRF2, a transcription factor that turns on a suite of endogenous antioxidant and detoxification genes. When NRF2 is turned up, cells are better equipped to neutralize reactive oxygen species and to reduce the oxidative triggers that would otherwise call the demolition crew, the MMPs, into action. Mechanistically, this matters because antioxidants that rely solely on being consumed can be transient. Activating NRF2 encourages your own cells to produce longer-lasting protective enzymes, like glutathione-related systems, shifting tissue from a reactive, damage-prone state to a maintenance-oriented state. In practical terms, that means broccoli helps both by reducing the signals that accelerate collagen loss and by improving the cell's ability to manage stresses that would damage matrix proteins. Practical tip: to maximize sulforaphane, lightly steam broccoli or eat a portion raw and include broccoli sprouts in your rotation. Sprouts can contain many times the glucoraphanin of mature florets. If you're using frozen broccoli, allow thawing and gentle cooking to preserve enzymatic activity. Overcooking destroys myrosinase and reduces sulforaphane formation. Some people use powdered broccoli sprout concentrates standardized for sulforaphane precursors when fresh options aren't available. Quick qualifier: activating NRF2 is a powerful protective strategy, but it's one piece of a broader plan. You still need amino acids, vitamin C, and an anti-inflammatory milieu for fibroblasts to build resilient collagen over time. 1. Sea buckthorn, the unusual oil that integrates with your skin. Sea buckthorn is a small berry with an outsized biochemical resume. It contains palmitoleic acid, a rare omega-7, various carotenoids, tocopherols, and a unique array of polyphenols. Palmitoleic acid is particularly interesting because it's a fatty acid that can integrate into cell membranes and skin lipids, supporting barrier function and hydration from the inside out. Improved barrier and membrane composition reduce transepidermal water loss and create a milieu where the extracellular matrix can maintain elasticity and function. Mechanistically, sea buckthorn's lipids help stabilize the physical environment that supports collagen and other structural proteins, while its antioxidant profile reduces the oxidative and inflammatory triggers that activate MMPs. In some clinical contexts, oral sea buckthorn and topical preparations have been associated with measurable improvements in skin hydration and elasticity. outcomes that align with a role in both membrane integration and extracellular protection. Practical tip: sea buckthorn is most commonly used as an oil, puree, or juice. For skin and barrier support, look for cold-pressed oils with minimal processing. For systemic antioxidant benefits, whole berry purees or high-quality juices preserve the broad matrix of carotenoids and polyphenols. Because its lipids are a major part of the activity, combining sea buckthorn with dietary fats or using it in dressings and smoothies helps integrate those compounds. Quick qualifier: sea buckthorn supplies unique fatty acids and antioxidants that support barrier function and hydration, but it's complementary work. It doesn't instantly rebuild collagen that has already been lost. Instead, it helps create and preserve the environment where rebuilt collagen can be functional. Synthesis: how these plants act together across the collagen life cycle. We now have the full countdown. Taken together, these 13 plants act across 4 interlocking domains: supply, signaling, protection, and structural stabilization. Supply means bringing in raw materials and essential cofactors, most centrally vitamin C from guava, acerola, amla, rosehip, and red bell pepper, so fibroblasts can carry out the chemical steps required to assemble stable collagen. Signaling means nudging fibroblasts to be productive and calming the hormonal or stress-based messages that tell them to stop. That's where ginseng and certain polyphenols play a role. Protection involves antioxidant and anti-inflammatory actions that blunt MMP activation. Think turmeric, green tea, rosemary, broccoli-driven NRF2 activation, and grape-derived proanthocyanidins. Structural stabilization covers the molecules that help collagen fibers resist breakdown and remain functionally cross-linked. Proanthocyanidins from grape, the polyphenolic scaffolds of amla and rosehip, and the membrane-friendly lipids of sea buckthorn. Aloe vera contributes hydration and mucopolysaccharide support that make the extracellular matrix receptive to properly formed collagen. The point is not that one plant will do it all, but that a dietary pattern rotating these botanical supplies, both the raw ingredients and the biochemical environment fibroblasts need. In a single meal, you might feed the enzymes, vitamin C, blunt an oxidative spike, EGCG from green tea, and provide a lipid context for absorption, sea buckthorn or a drizzle of olive oil. Over time, repeated meals build a persistent tissue environment where collagen synthesis outpaces breakdown more consistently. Practical roadmap: choose 4 to 6 staples and rotate them weekly. To turn this into a manageable habit, pick a small core set you enjoy and can access reliably. Here's an example of a simple weekly rotation that balances convenience with mechanistic breadth. Core staples: choose at least 4. A vitamin C source, guava, acerola, or amla. A daily tea, green tea, catechin habit. Turmeric used in cooking, a grape-based polyphenol source. Whole grapes or grapeseed extract. Broccoli or broccoli sprouts, and a lipid-rich plant like sea buckthorn oil or culinary oils paired with carotenoid-rich plants. Red pepper. Sample microplan. Day 1, vitamin C focus. Morning smoothie with frozen guava or freeze-dried acerola, a scoop of protein, and a tablespoon of nut butter. Snack on raw red pepper. Evening green tea. Day 2, anti-inflammatory herbs. Turmeric-rich curry or turmeric vinaigrette with black pepper and olive oil. Roast rosemary seasoned vegetables. Broccoli sprouts added to a salad. Day 3, polyphenol stabilization. Mixed berries and red grapes for breakfast. Grapeseed extract or a handful of nuts with dark chocolate as a snack. Sea buckthorn oil blended into a yogurt dressing. Day 4, hydration and polysaccharides. Oral aloe fillet gel, quality inner fillet product, or a fresh aloe-based smoothie addition paired with vitamin C-rich fruit. Day 5, hormonal resilience support. A cup of Korean ginseng tea or a modest standardized extract if that fits your health profile. Pair with a vitamin C snack to support synthesis. Day 6. NRF2 activation. Lightly steamed broccoli and broccoli sprouts added to meals. Include green tea. Day 7. Flex Feast Day. Combine several elements. A vegetable-forward meal with red peppers and rosemary, a berry-rich dessert, and a green tea. Rotate and repeat. If budget or palate limits you, prioritize vitamin C-rich foods plus at least one MMP modulating plant, green tea or turmeric, and one stabilizer, grapeseed or sea buckthorn. The most important variable is consistency, daily or near-daily inclusion of those signalers and cofactors, bioavailability and preparation tips that matter. Turmeric. Pair curcumin with piperine, black pepper, and a dietary fat to enhance absorption. Alternatively, consider standardized curcumin extracts, for example those standardized to 95% curcuminoids, when looking for research-like consistency. Carotenoids, red bell peppers, sea buckthorn. Consume with a source of fat to improve absorption. Light cooking can liberate carotenoids from plant matrices. Vitamin C matrix, amla, acerola, guava, rosehip. Whole fruit or freeze-dried whole fruit powders preserve polyphenol scaffolds that appear to stabilize vitamin C compared with isolated ascorbic acid. Broccoli, sulforaphane. To maximize sulforaphane, chop raw and allow to sit briefly before cooking. Eat lightly steamed rather than boiled. And include broccoli sprouts when possible. Or consider sprout concentrates standardized for glucoraphanin when fresh produce is unavailable. Grape proanthocyanidins. To mirror research contexts, grapeseed extracts standardized for proanthocyanidin content are the form commonly studied. Otherwise, prioritize whole grapes, skins and seeds when possible for dietary polyphenols. Aloe vera. Choose inner fillet oral products or freshly prepared inner gel rather than whole leaf products that include latex. Avoid added sugars or syrups in commercial formulations. Extract versus whole food. Whole foods give a matrix of cofactors and fiber that supports absorption and reduces spike-type exposures. Use standardized extracts when research suggests a concentrated active ingredient is needed, or when access to fresh whole foods is inconsistent. Look for transparency in manufacturing, clear standardization of the active fraction, when relevant, and minimal unnecessary additives. Safety notes and caveats. General population. These plants are food-forward and safe for most adults when included in normal dietary amounts. However, concentrated extracts can have interactions or contraindications for certain people. Within pregnancy, breastfeeding, and medication interactions, the transcript does not provide exhaustive safety profiles, so specific guidance for these groups is indefinido. If you are pregnant, nursing, or taking prescription medications, especially blood thinners, blood pressure drugs, or hormone therapies, consult a qualified healthcare professional before beginning concentrated herbal extracts. Quality matters. Some commercial products contain fillers, added sugars, or non-standardized active fractions. Favor companies with third-party testing, clear extract standardization, where applicable, and transparent ingredient lists. No plant is a substitute for medical care. If you have a medical condition affecting connective tissue, skin, or joints, these botanicals are supportive adjuncts and not replacements for prescribed therapies. What instant really means in practice? Returning to the title, the word instantly can mislead if taken to imply complete structural reversal in a single moment. We keep that promise creatively and accurately by emphasizing the real, immediate biochemical effects these plants can have. Within hours of consumption or topical application, you can see measurable changes at the molecular level. Antioxidant capacity rises, MMP signaling can be blunted, cofactor availability changes, and cellular stress response pathways like NRF2 begin to alter gene expression. Those are real and meaningful immediate effects. They reduce ongoing damage and set the stage for repair. Visible, reliable increases in tissue firmness, wrinkle depth reduction, or recovery of severely degraded collagen require time. Most people notice incremental improvements in skin hydration and a sense of resilience earlier, while structural rebuilding of the dermal matrix generally occurs over weeks to months of consistent support. That's why the best approach is both immediate, include antioxidant and cofactor-rich plants now, and sustained, rotate and repeat them daily or weekly. Measuring progress. Practical markers to watch. You don't need lab tests to detect benefit. Practical, consumer-accessible markers include improved skin hydration and luminosity, reduced dryness and irritation, fewer days where sun exposure causes prolonged redness, and subjective improvements in elasticity or joint comfort for some people. Photos taken consistently under similar lighting over weeks can help track visible changes. For more objective measures, some people monitor hydration scores from consumer skin analyzers or seek professional dermatological assessments, but these are optional and not required to benefit. Choosing products and reading labels. When a concentrated extract is appropriate, look for clear standardization, e.g., grapeseed extract standardized for proanthocyanidin content, curcumin standardized to 95% curcuminoids. Third-party testing results or certificates of analysis. Minimal filler ingredients and transparent sourcing statements. Whole fruit or whole plant designations when the polyphenolic matrix is part of the mechanism. Rose hip, amla, acerola. Final practical checklist to start today. Pick 4 staples. A vitamin C source, guava, acerola, amla. A green tea habit, turmeric prepared with fat plus black pepper, and a broccoli or broccoli sprout inclusion. Add 1 stabilizer, grapes, grapeseed or rose hip. Add 1 lipid support item, sea buckthorn oil or a healthy culinary oil to improve carotenoid absorption. Aim for small, repeatable actions. A daily smoothie with vitamin C, a cup of green tea, turmeric in a cooked meal 3 times a week, and broccoli at least twice weekly. Rotate to cover different mechanisms and preserve variety. Closing, reclaiming an inside-out strategy for resilient collagen. If you've been treating skin and connective tissue as a surface-only problem, this countdown reframes the fight. The meaningful battlefield is cellular. These 13 plants don't promise an overnight miracle, but they do offer immediate molecular-level defenses and repeated cumulative signals that allow fibroblasts to do what they were built to do: synthesize and maintain resilient collagen. Start with a few staples, pair them thoughtfully for absorption, and rotate them so your cells receive a steady stream of cofactors, anti-catabolic signals, and stabilizing polyphenols. If you want a printable checklist of the 13 plants and easy weekly plans for how to use them, find the link in the description. If this evidence-based approach to nutrition and anti-aging sounds useful, subscribe. We'll keep breaking down the research and turning it into practical habits you can start this week.